Hello, students. Today we begin an essential and fascinating topic in microbiology, the morphology and classification of bacteria. Understanding how bacteria are structured, how they appear under the microscope, how they grow, and how they are classified is fundamental to microbiology. If we cannot recognize bacteria, how can we identify them, control them, or treat infections caused by them? Build a strong conceptual foundation step by step. Let us first understand where bacteria stand in the world of microorganisms and how microbiologists group these tiny life forms for study. Microorganisms are a heterogeneous group. Based on cellular organization and biochemical differences, they are divided into two major groups, prokaryotes and eukaryotes. Bacteria belong to the prokaryotic group. Prokaryotic cells are structurally simpler than eukaryotic cells and do not have a nucleus enclosed by a nucleomembrane. Instead of a true nucleus, bacterial genetic material sits in a nucleoid region. Bacterial morphology starts with shape. The three classic shapes are Cocci, Bacilli, and Spirilla, meaning spherical, rod-shaped, and spiral respectively. Arrangements describe how cells associate. Pairs, chains, clusters, and other groupings form depending on division planes and growth patterns. Examples include diplococci in pairs, streptococci in chains, staphylococci in grape-like clusters, and bacilli appearing singly or in chains. Special shapes include vibrio, fusiform, and filamentous bacteria. Shape influences motility, nutrient uptake, and where bacteria thrive in the environment. Now let's break down a typical prokaryotic cell. Cell wall, plasma membrane, cytoplasm, ribosomes, and the nucleoid region containing DNA. Cell walls differ. Gram-positive bacteria have a thick peptidoglycan layer, while gram-negative bacteria have a thinner layer plus an outer membrane. Capsules, pili, and flagella are external structures that aid protection, attachment, and movement. The nucleoid is not membrane-bound. It is an irregular region where the chromosome is concentrated and often anchored to the membrane. Plasmids are small circular DNA molecules that can carry antibiotic resistance or metabolic genes, influencing clinical outcomes and classification. Ribosomes in bacteria are 70S and differ from eukaryotic 80S ribosomes, a difference exploited by certain antibiotics to selectively target bacteria. Observation begins with light microscopy. We'll demonstrate bright field and phase contrast images to reveal shape, size, and arrangement. Simple stains like methylene blue reveal morphology and size, while differential stains provide more diagnostic information for classification. Gram staining uses crystal violet, iodine decolorization, and safranin. Gram-positive cells retain violet, while gram-negative cells take the counterstain. Electron microscopy reveals ultrastructure such as flagellar motors and envelope layers not visible by light microscopy, useful for advanced study. Culture appearance, biochemical tests, and metabolic profiles complement morphological and staining data to build a reliable identification of bacteria. Classification combines morphology, staining results, physiology, biochemical traits, and genetic data to assign bacteria to genera and species. Modern methods increasingly use 16S RNA sequencing to determine evolutionary relationships with higher resolution than morphology alone provides. Despite advances in genetics, morphology remains important for rapid clinical decisions, environmental surveys, and initial laboratory assessment. Understanding bacterial structure helps prevent and treat infections, guide antibiotic selection, and design diagnostics and public health responses. Summary: learn to recognize basic shapes and arrangements, understand cell structures like the nucleoid, and combine staining plus genetic tools for classification. Thank you. Keep practicing microscope skills, review labeled diagrams and animations, and build on this foundation for all future microbiology learning. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, Connecticut, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.